This is the first video in this series in which we will discuss and analyze the numerous psychological studies covered in the Cambridge AS Psychology syllabus. In this series, we'll discuss how several of the most famous and influential experiments in history were conducted in order to understand how and why the researchers came to their conclusions. But we will also make an effort to look at how an understanding of those experiments can be applied to understanding our world, to look at why these experiments were considered sufficiently interesting and insightful to be included in the syllabus. Let's get started. Emotion, Memory, and the Amygdala, Canli et al. 2000. Have you ever noticed that you were better able or more likely to remember events in which you experienced a strong emotion? Have you ever wondered whether this phenomenon was ever studied, or if there were any scientific explanations for this? It might surprise you to know that it has been, and there is. One of the studies that demonstrated the relationship between memory and emotion most clearly was done by Turhan Canli and 4 of his associates in 2000, titled Event-Related Activation in the Human Amygdala Associates with Later Memory for Individual Emotional Experience. Let's look at that experiment to understand and analyze the brain structure being measured, the process by which it was measured, how the study built on previous research, the aims of the study, and its research methods and design. Finally, we'll look at its results and conclusions, and we'll briefly discuss the study's potential strengths and weaknesses. Before we get started, ask yourself, how much do you think the formation and retention of long-term memories is associated with emotion? Do you find that your own most salient memories have been formed during or following highly emotional events? If you have noticed this phenomenon, why do you think that is? Hopefully, by the end of this video, you'll be able to see if your preconceived understanding of reality matches with the psychological research on this phenomena. The amygdala, what is it? This experiment studied the role of a brain system called the amygdala in forming emotional memories. The amygdala is a system of 2 almond-shaped brain structures on the left and right sides of the brain. Its full functions haven't been fully explored, but its activation has been shown to be associated with processing emotions and with storing and recalling emotional memories. Early research focused on the amygdala's role in processing negative emotions, but more recent research has found that it also plays an important role in processing positive emotions and emotional memories. There were 2 main goals of Canli et al. 2000. 1, to study the degree that amygdala activation is involved in the processing of emotionally intense images, and 2, to study the degree that emotional intensity affects the role of the amygdala in enhancing the memory formation and the recall of emotionally intense stimuli. In other words, the goals of Canli et al. Were to demonstrate and measure how highly emotional images are remembered better than those with little emotional impact, and how stimuli which produce greater amygdala activation might be recalled more easily than stimuli that generate less amygdala activation. The research team aimed to study the degree that the amygdala is sensitive to varying degrees of emotional intensity, and whether the activity of the amygdala is correlated with enhanced memory retention. Before going on, take a second and consider, what would your own hypotheses be? To what degree does the emotional intensity of events affect the formation and retention of memory? And to what degree do you think the amygdala might be part of this experience, based on what you know so far? Functional magnetic resonance imaging, or fMRI. Before describing the study, we should look at the main diagnostic tool it employed. Improvements in technology have made it possible to observe the brain activities of people as they occur. fMRIs work by measuring changes associated with differences in blood flow, with the assumption that changes in blood flow correlate with changes in activity in different areas of the brain. fMRIs are often used to measure changes in brain function between different activities. To do this, participants have their brains scanned in a rest state to establish baseline activity, then scanned again during or shortly after completing an experimental or control task. If the fMRI finds patterns of differences in brain activity between resting, control, and experimental groups, The changes are inferred to be caused by the differences in brain functioning caused by the differences in those activities. Previous studies. Science is not done in a vacuum. Nearly all scientific studies occur in response to previous studies to find or clarify something that was not made clear in previous research. This study by Canli et al. Is not different. In this case, Labar and Phelps in 1998 found evidence that emotional experiences are often better recalled than non-emotional experiences, and that emotional arousal increases the likelihood that short-term memories will be consolidated as long-term memories. If you look at this graphic of memory formation, in which sensory input is registered by some part of the consciousness, then stored as short-term memory, and finally, through a process of internal rehearsal stored as long-term memory, emotional arousal would increase the probability that a memory would be stored as a long-term vivid memory. Brain imaging studies have confirmed that amygdala activation correlates with the formation and recall of emotional memories. But a previous study by Canli et al. In 1999 found that there could have been confounding variables present in those studies, which casts doubt on their validity. The study we're looking at in this video by the same team done the following year was designed to test this. Variables. This was a laboratory experiment. The independent variable, which is the variable being manipulated, was the series of 96 emotional images which were shown to the subjects over the course of the experiment. The published experiment discusses the independent variable as the emotional intensity of those images, rather than the images themselves. Phases 1 and 2 of the experiment had two dependent variables each, which are the variables being measured that changed in response to the subjects viewing the images. In the first phase of the experiment, the researchers measured two dependent variables: self-reports of participants' emotional responses to the images on a scale of 0 to 3, and the participants' simultaneous brain scans, in which the fMRI machine measured participants' relative amygdala activation during and following exposure. In the second phase, the researchers measured participants' ability to recall images based on their previous emotional responses to those images, while also measuring participants' amygdala activation to see if greater activation correlated with greater ability to recall those images. Sample. The sample was 10 right-handed healthy female volunteers. Research has shown that there are differences between the brains of men and women and between left and right-handed people, and the researchers clearly wanted to avoid these differences as confounding variables. Women were exclusively used in this study because it was believed that women are more likely to report intense emotional experiences and to show more physiological reactions to emotional stimuli. The idea that women are more emotional than men is a stereotype in Western popular culture, but evidence for this has been found so frequently in psychological research that it has come to be regarded as a statistical truism. In general, psychological research shows that women at a statistical level tend to demonstrate greater emotional expression than men, experience emotions more intensely, report more comfort in expressing emotions, and tend to be better at reading nonverbal cues than men. So, the researchers' decision to use women to study emotion wasn't based on any bias that was unsupported by the scientific literature. The vast majority of people are right-handed, so the participants' decision to use right-handed people both likely made it easier to find participants and made the results of the study more widely generalizable to the general public. Procedure. Stage 1. This experiment was conducted in 2 stages. In the first stage, a series of 96 images were shown to participants from an overhead projector and mirror while they were laying down in an fMRI scanner. All participants were given informed consent and were aware of what they had to do. The 96 images were chosen from a set of images called the International Affective Picture System, a standardized set of images which is often used in psychological experiments. These are some examples of images used in the image set, representing awe, excitement, anger, sadness, contentment, amusement, disgust, and fear. The order of the pictures was randomized between participants, so the order of the images was not a confounding variable. Participants were told to view each image for 12.96 seconds and then press one of 4 buttons to indicate their level of emotional intensity as they witnessed each of the scenes while inside the fMRI machine. The numbers ranged from 0, which represented not emotionally intense at all, to 3, which represented extremely emotionally intense. The fMRI recorded participants' amygdala activation to measure how closely the activation of the amygdala correlated with subjective emotional responses. Procedure, Stage 2. 3 weeks after the first part of the experiment, the participants received an unexpected call to return to the laboratory and participate in the second phase of the experiment. In the second task, the participants viewed the 96 images again, plus 48 new images called foils. The foils were selected to match the previous image's emotional arousal characteristics, as had been defined in previous research. Participants were asked whether they had seen each scene before. If the participants responded that they had, they were asked whether they remembered with certainty, called remembering, or whether they had a less certain feeling, called knowing. Again, the fMRI recorded participants' amygdala activation to measure how closely this correlated with the participants' ability to recall any of those images. So, what were the results? Results, Stage 1. Participants' ratings of emotional intensity correlated well with typical subjects' ratings in other experiments, demonstrating that the pictures were effective at eliciting appropriate emotional reactions from the participants. The study found that greater amygdala activation was significantly correlated with higher ratings of self-reported emotional intensity. This graph shows the clear relationship between subjective emotional intensity and amygdala activation that was observed in the experiment. Results, Stage 2. The follow-up memory task indicated that memory performance was significantly improved for scenes that were rated as highly emotionally intense. That is, rated 3 on the scale. Then for scenes rated as less intense, fewer highly emotionally intense scenes were forgotten and more were remembered than was common for less intense images. A second result of the follow-up task was that for scenes that were rated as highly emotionally intense in the first stage, amygdala activation for the left, but not the right, amygdala predicted whether stimuli would be forgotten, seem familiar, or be remembered in the second stage. In cases where the amygdala activation and reported intensity did not match, it was the amygdala activation and not the reported emotional intensity that was correlated with later recall. The experiment also found that the activation of other brain structures correlated with different portions of the experiment, supporting the idea that the amygdala functions as a coordinated part of several brain systems and not alone. Unfortunately, because these other brain structures were not the focus of this experiment, they were not published. Strengths and weaknesses. Since this was a lab experiment in which all participants were tested in a standardized environment and given the same items to rate in each condition, the experimenters eliminated all obvious confounding variables, and the study had good internal validity. The researchers' use of an fMRI machine gave the researchers a lot of quantitative data related to the function of the amygdala, so they were able to carry out detailed statistical analyses following the experiment. However, the location of many specific behaviors in the brain is still not well understood, and this makes it difficult to come to definitive conclusions about how the data can be applied to specific behaviors. Since the experiment was done in a highly artificial setting, this could reduce the ecological validity of the experiment. That is to say, the phenomena studied in this experiment might occur differently in the real world to the degree that actively being measured by an fMRI machine might make the brain react to stimuli differently. Because the study only sampled right-handed females, the sample population could lower the validity and reliability of the experiment. That is to say, it might be difficult to generalize the results towards non-right-handed people or men. And at the time of this study's publication, more research needed to be done to confirm this. The sample size was quite low, though subsequent studies have since replicated this study's results. Some findings were only correlational and not necessarily causal, such as those finding an association between emotional impact and later recall. The correlational nature of the data potentially casts doubt on any causal explanation for events that occurred in this experiment. Conclusions. What can students or the general public learn about life from this experiment? 1. Amygdala activation is highly associated with individuals' subjective emotional intensity. Activation of the left amygdala was especially predictive of later recall for reasons which were not clear in this experiment. 2. The more emotionally intense an event or experience is, the better it will be remembered. This correlates with and confirms many people's subjective experiences. How well do the conclusions of this experiment match your own subjective experience of the world? Let me know in the comments. Thank you for watching. I'm going to continue this series on psychology in the future, so please subscribe for future content. In our next video in this series, we will discuss an experiment studying the relationship between REM sleep and dreams.